Welcome viewers, welcome to the new lesson from Dr. R's Biology. If you did not subscribe the channel, you can just click on the subscribe button and bell icon. Welcome students, in this particular lesson we will be just learning about the Krebs cycle, where you can see the complete oxidation of glucose molecule happens. Clear? So let us see what are the different steps in glycolysis. Before that, you have to just watch the briefing of this particular Krebs cycle. You can just watch that. It's here now. So it is better to see the Krebs cycle in brief before you going into the details of each and every steps in Krebs cycle. So here we can see that Krebs cycle, the complete pathway of chemical reaction in Krebs cycle was elucidated by Hans Krebs, and that's why the name Krebs cycle. And it is a cyclic pathway where the acetyl coenzyme A enters the cycle along with the oxaloacetate and leave as CO2, and it will be retrieving the oxaloacetate back. It is otherwise known as citric acid cycle, CAC cycle, because the first product which is resulted due to the combination of acetyl coenzyme A and oxaloacetate is citric acid. It is otherwise known as tricarboxylic acid because the citric acid, the first product of Krebs cycle, is a tricarboxylate because it is containing 3 carboxylic acid group, COOH group. The Krebs cycle will be taking place in the mitochondrial matrix in contrast to the glycolysis which proceeds in cytoplasm. Now let us see what are the steps in Krebs cycle. Here we can see that in this particular figure we can see different steps in Krebs cycle. First step, the acetyl coenzyme A will be combining with oxaloacetate resulting in the formation of citrate. This is catalyzed by the enzyme citrate synthase, and you can see an addition of water molecule for this combination process. Now the citrate will be converted into isocitrate, and it will be through the formation of an intermediate compound known as cis-aconitate. Here the citrate, the water will be removed from it and then the water will be added to form the isocitrate. So that's why the cis-aconitate intermediate is there. As this intermediate is there, the enzyme that catalyzes this reaction is known as the aconitase. Now the isocitrate is undergoing an oxidative decarboxylation process where the isocitrate will get oxidized, where NAD plus will be acting as the oxidizing agent, and also the carbon dioxide will be removed. Here in this step, NADH plus H plus molecules will be released out. As we know from the glycolysis itself, there are 2 types of cycle is happening. 2 times the cycles will be repeated because 2 molecules of pyruvate is formed as a result of glycolysis. So here also you have to expect the cycle is repeating 2 times, and as a result 2 NADH plus H plus will be formed here. Now the product formed will be alpha ketoglutarate, where isocitrate dehydrogenase is playing a role. Now the alpha ketoglutarate again undergo one more oxidative decarboxylation. As a result, 2 molecules of NADH plus H plus will be formed along with the release of one CO2 molecule. The product formed from this reaction is succinyl coenzyme A, and it will be catalyzed by the enzyme alpha ketoglutarate dehydrogenase. Now the succinyl coenzyme A will be just removing its coenzyme A part, and as a result, succinate will be resulted. The removal of succinyl coenzyme, that coenzyme part of the succinyl coenzyme A, will be releasing some energy, and that energy will be utilized to combine GDP plus orthophosphate to form the GTP. Now, the enzyme catalyzing this reaction is known as the succinyl coenzyme A synthetase. This succinate is then undergo another process, and as a result, fumarate is formed. And it will be a dehydrogenation process or another redox reaction. Here the redox potential is comparatively lesser, and as a result FAD will be playing a role as oxidizing agent, and FADH2 will be resulted. Now the fumarate is converted into malate by the addition of one water molecule by the enzyme fumarase, and this is a class of lyase enzyme. As a result, malate is formed. Now this malate undergo dehydrogenation process or the redox reaction, or we can say the oxidation process, and as a result NADH plus H plus will be released with the retrieval of oxaloacetic acid, which was first involved by in combination with acetyl coenzyme A. Now here we can see that up to the formation of succinyl coenzyme A, there is the oxidation of the glucose happening, and after that particular step is just the retrieval of the oxaloacetate. Okay, so one decarboxylation is happening here, and another decarboxylation is happening here, and one decarboxylation is happening in the formation of acetyl coenzyme A. So 3 decarboxylation 2 times results in the complete oxidation of glucose, and as a result the energy will be released. And the remaining steps in glycolysis, as I marked, it is just the retrieval of oxaloacetate. So keep in mind that the steps which are forming the NADH and GTP plus FADH2. Now let us see the details of Krebs cycle. Each and every steps will be explained now. Okay, then you have watched it, so we can see that the Krebs cycle is actually the cyclic process where the complete oxidation of glucose took place with the concomitant release of the 4-carbon molecule, which was actually entered into the Krebs cycle. Okay, so let us see each and every steps in detail so that you can write the essay question easily. Okay, why I have given the briefing first is, if you are having a good idea what is going to happen, then it will be easier for you to learn each and every step. Okay, so let us see the steps. We know that actually the Krebs cycle, the pyruvate is entering into the Krebs cycle by conversion of it into acetyl coenzyme A, that we have learned when we learned about the fates of pyruvate. One more thing, make sure that before reaching the Krebs cycle part, you have to ensure that you have already covered the glycolysis part and fates of pyruvate. Okay, from the fates of pyruvate, aerobic fate, we know that the pyruvate is converted to acetyl coenzyme A. Acetyl coenzyme A, and this acetyl coenzyme A is formed by the action of pyruvate dehydrogenase complex enzyme. Pyruvate dehydrogenase complex enzyme, where you can find the formation of NADH plus H plus NADH plus H plus, and the involvement of coenzyme A with the release of CO2. So this happened there in the conversion of pyruvate to acetyl coenzyme A. Now this acetyl coenzyme A is taking part in the Krebs cycle, is entering into the Krebs cycle. Okay, so the first step of Krebs cycle is the union of a 4-carbon acid with a 2-carbon acid forming a 6-carbon acid. And that 6 carbon acid is nothing but the citric acid, which is otherwise. That's why the Krebs cycle is otherwise known as the citric acid cycle. This particular citric acid is actually containing 3 carboxylic acid group. And as a result, the Krebs cycle is also named as tricarboxylic acid cycle, TCA cycle. So it is known as citric acid cycle, CAC. It is also known as tricarboxylic acid cycle, TCA cycle. And it is known as Krebs cycle also. Okay, so hope you got why this particular name came. So in this, the oxaloacetate will be coming. So let us see. You have to keep in mind one more thing, that when you draw the reactions, the carboxylic acid, the Krebs cycle should be in this way. Okay, so acetyl coenzyme A is entering, some cyclic reactions are happening, and something is releasing out. It will be like this. So I will be just drawing in the linear form only, but make sure that you are adding each and every component in the circular fashion. In the form just we have watched the briefing, right? In the same pattern. I will be just describing each and every step in this way only, not in the circular form. As it is a cyclic process, we have to represent this in this form. Okay, so ensure that you are drawing the structures and reactions in this way only. If you want to know it, you just go back to the video, initial part of the video, and just see that particular image. Okay, so acetyl coenzyme A is now combining with the oxaloacetate. Okay, oxaloacetic acid is actually a tricarboxylic acid. What is meant by dicarboxylic acid? That means it contains 2 carboxylic acid group. Okay, so let us see that. So it is nothing but the COO minus CH2. Okay, now CHOH, C double bond O, and COO minus. So this is what actually the oxaloacetic acid. Now, this particular oxaloacetic acid will be combining with the acetyl coenzyme A. I am just representing acetyl coenzyme A in a different color so that you will understand where this joining is happening. So, I am just representing acetyl coenzyme A. We know that acetyl coenzyme A is CH3, C double bond O, SCO-A. Okay, now as a result of this combination, what happens is every reaction is irreversible, not irreversible, reversible only. So I will be just representing the single arrow mark only, but make sure that you are taking it as the reversible reaction. As no reactions in the Krebs cycle is irreversible, we are just representing with a single arrow marker. Okay, if both kind of reaction is happening in a particular cycle, you can just represent with a double arrow mark and a single arrow mark to differentiate the irreversible and the reversible reaction. As all the reactions of Krebs cycle are reversible, I am just using single arrow mark only, nothing else. We don't have to differentiate it actually. So we can see that the citric acid is formed. So when citric acid is formed, what will be released out? Definitely this particular SCO-A should be released out. So we can see that SCO-A is released out. Okay, but there is an addition of something. A water molecule will be added. So I am just writing HOH. HOH. Do you think that this particular HOS, that is water, is coming from outside? Definitely no. It is coming from one of the steps in glycolysis. Which was that particular step? The formation of phosphoenol pyruvate. Actually, that water is this particular thing. There is no extra addition of any compound. Instead, it is already there. It is actually released from the glucose only. So HOS, that was released in phosphoenol pyruvate formation, don't forget that, is added. So let us see what is happening here. So HS square should be released out. HS square. So you know this hydrogen is actually this. So this will be cleaving this particular molecule. Okay, this particular thing will be just cleaved. Now we can see that this CH3, CH2H, you can just take it like that. This will be getting attached to this particular portion, and as a result you can find this CH2CO. In that way it will be going. So let us see that particular thing. C double bond O, and you will find the water molecule OH. I'm just writing it with the different colors so that you will understand from where these and these are coming. And now this CH3. Okay, so this CH3 will be just CH2. Okay, now what will be happening? This will be attaching to this particular carbon. This will be attaching to this particular carbon. As a result, what will happen? This bond will be breaking. Only single bond O will be there. So O minus comes here. Now the one hydrogen that is remaining there will be coming and binding here. Got it? So we can see that the CH2, then it will be coming down COO minus. Remember, this CO minus is actually COH only. These are deprotonated or ionized state of the carboxylic acid. And here CO and H is coming from the acetyl coenzyme A, so H. Got it? And now the remaining portion will be exactly same. CH2 COO minus. This is the thing. So where we have added the water molecule, it is actually hydrolyzing. It is actually hydrolyzing the acetyl coenzyme A into the acetyl part and coenzyme A part. So OH will be added to this carbon, H will be added to this sulfur. That's why the sulfur HS square is released. Now OH, then it becomes the carboxylic acid group. So you can see that there are 3 carboxylic acid group in this. So this is the first one, this is the second one, and this is the third one. As it is 3 carboxylic acid group containing molecule, it is known as tricarboxylic acid. So citric acid or citrate, that is formed in the first step of Krebs cycle, is tricarboxylic acid, and therefore the Krebs cycle is also known as tricarboxylic acid cycle, TCA cycle. It is citrate, therefore it is known as citric acid cycle. Hope it is clear for you. Now what is the enzyme that catalyzes this particular reaction? It is nothing but the citrate synthase. Citrate synthase. Now there comes another confusion. You will be just seeing synthase and the synthetase enzyme. Both will be actually catalyzing the synthesis of molecules. Then what is the difference between synthase and synthetase? It is nothing but the involvement of nucleotide triphosphates. If ATP, GTP, CTP, UTP or TTP, anything is involved in the synthesis of any molecule, it will be synthetase enzyme. If nucleotide triphosphate energy is released and the synthesis happens, it will be synthetase enzyme. So you can just see T, ATP, T, synthetase T. If ATP is not involved, if nucleotide triphosphates are not involved, it will be synthase enzyme, and that's why it is citrate synthase. We know that there is no energy releasing nucleotide triphosphate involved in this reaction. Now the question is, actually synthesis need energy. Then why no energy provider like ATP is here? Answer to the question is, we know that acetyl coenzyme A contain coenzyme A. The removal, hydrolytic removal of coenzyme A will be releasing energy. Okay, that can favors a reaction which, if on otherwise that particular reaction is thermodynamically unfavorable. Okay, so this release is actually, this removal of the coenzyme A is actually releasing energy which can favor the reaction. Okay, so that's why, so hope you remember whatever I said. Okay, why synthase enzyme name, why it is not synthetase, why energy releasing compounds are not there. Okay, so citrate. Now let us see the second step. So I am just taking it back, but you and remember that you have to make it in a circle like this. It should go like that. Okay, but I am just drawing in order to, as I don't have enough space here, I am just drawing it in the line only. But you have to make sure that you are just representing it with the cyclic form. Okay, so now let us move to the next step. In the next step, citrate is actually isomerized to isocitrate. Citrate is isomerized to isocitrate. Now I am not using any color. Okay, so I am just leaving the color because this color I have used only to represent which part of acetyl coenzyme A and which part of oxaloacetate is joining. So I am just using the single color. Okay, I will use the black color. It will be just converted into as isocitrate. From the name itself, what will be the enzyme? It will be isomerase enzyme. Here, the name isomerase enzyme, name of the isomerase enzyme is aconitase. Aconitase. Why aconitase? Because in between citrate and isocitrate, there is an intermediate compound known as cis-aconitate. So a cis-aconitate is there. Okay, cis-aconitate is there, where that is actually converted into isocitrate. So in between citrate and isocitrate, cis-aconitate is there, and this presence is actually making the name aconitase. Now, how this aconitate will be formed? It will be formed just by removing the water molecule. Just by removing the water molecule, this aconitate will be formed, and just by addition of water molecule, isocitrate will be formed. The difference between citrate and isocitrate is this OH will be just shifting to this particular point, and this hydrogen will be just shifting to this particular point. That's all. Okay, so let us see the citrate, isocitrate structure. COO minus, I'm just doing this particular thing, CHOH, CHCOO minus, CH2COO minus. So this hydrogen, this OH just shifted from one place to another, and as a result the isocitrate is formed. Sometimes you can see in some of the textbook this H and this OH is changed. There is no issue in that because you can see that it is almost the same structure only. You are just turning it. Okay, so you don't have to worry about it. If you are finding any OH here and H there, just turn the molecule, that's all. Clear? So don't get confused when you are seeing that particular diagram. So that's about the second step of Krebs cycle. So citrate is isomerized to isocitrate, catalyzed by the enzyme aconitase. The name of the enzyme is coming from the intermediate compound cis-aconitate, which is actually the product which is formed by the removal of water from the citrate. When 6-aconitate, the water is added, it will form isocitrate. But OH and H position will be varying. So, in order to show the removal and addition of water, we have to just mark it like this. So, water is removed, then water is added. Okay, so water is removed and water is added. So, you have to just mark it like that. As a result, isocitrate is formed. Now, let us see the next step where isocitrate is converted into alpha-ketoglutarate. So that is the second oxidative decarboxylation happening to glucose. Okay, the first one is there in this itself. Pyruvate is converted into acetyl coenzyme A. Now we are just moving into the second oxidative decarboxylation. Okay, so let us see that. Isocitrate is now undergoing oxidative decarboxylation process. So we can see here, this particular thing we can see here, this particular carbon dioxide COO minus will be released out. This will be released out. But keep in mind, this also is happening in two-step process where an intermediate compound known as oxalosuccinate is there. Anyway, for degree program, you don't have to learn about that intermediate compound. That's why I did not draw the structure of 6-aconitate also. Okay, so but keep in mind that there is two-step process. That is, we are just representing it in a single step, but actually it is a two-step process where an intermediate compound oxalosuccinate is formed. It is because it will be happening like this. First, the oxidation will be happening to this particular molecule H or HH. This particular 2 hydrogen will be released out, and as a result oxalosuccinate will be formed. And now in the next step, this carbon dioxide will be removed. Then alpha-ketoglutarate will be formed. That is how the reaction process. But we are just representing it in a single step process. So we can see here isocitrate is undergoing oxidative decarboxylation. So the first step is oxidation. Okay, so as a result of oxidation, we can see that the NAD plus is now just forming or undergoing reduction by oxidizing isocitrate. It is forming NADH plus H plus H plus. Okay, so from where this came, this hydrogen and this hydrogen, so it will be C double bond O here then. Now what should happen? This particular carbon dioxide should be removed. So we are just showing that release of carbon dioxide. So this is actually removed. Okay, this particular carbon dioxide is removed. And what is the enzyme? It is nothing but dehydrogenase enzyme, because it's a kind of redox reaction where reduction is happening to NAD plus and oxidation is happening to isocitrate. So redox reaction is there. So it will be dehydrogenase enzyme. And what will dehydrogenase? Isocitrate dehydrogenase. Isocitrate dehydrogenase enzyme catalyzes the redox reaction along with the decarboxylation, and as a result, what will be formed? The alpha-ketoglutarate. So COO minus, and what is remaining here? H gone. So C double bond O is remaining. Now what is remaining here? This carbon dioxide has gone. Okay, this particular carbon dioxide has gone. So what is remaining here? CH. So what will be the bond? Again the H plus will be added. We know that this is ionized. This is actually COOH. Ionized to this one. When it is ionizing, there will be plus H plus, right? CO minus plus H plus. So that H plus will be added at this particular point. So we will be just adding that point H plus in that way. Okay, H plus will be added. So CH2. And what is remaining here? CH2. And then COO minus. This is actually the alpha ketoglutarate. So this is the alpha ketoglutarate. So azan time I will just come after the azan. Okay, so in this step isocitrate is oxidatively decarboxylated into alpha ketoglutarate with the formation of NADH plus H plus and release of carbon dioxide. So how many carbon dioxide or carbon is left in the glucose now? Only 2, because 2 we have lost here and 2 we have lost here. Why 2? 2 I just represented only 1. Why 2? Because we know that 2 molecules of pyruvate is involved. That's why 2 CO2 is removed and then 2 CO2 is removed here. So only remaining is the 2 carbon. So after the removal of that 2 carbon, and their glucose oxidation will be over. So let us see the next step. I am just drawing it in this way, but make sure that you are just drawing in the cyclic fashion. Clear? Don't forget what is oxidative decarboxylation and what are the 3 oxidative decarboxylation happening in terms of glucose oxidation. So let us see the next step. So alpha ketoglutarate. This alpha ketoglutarate is again undergoing one more oxidative decarboxylation. So one more oxidative decarboxylation is happening to alpha ketoglutarate. We know that alpha ketoglutarate has formed by oxidative decarboxylation. Now this molecule itself is again undergoing one more oxidative decarboxylation. So what will be the So carbon dioxide will be removed, and at the same time, what will be removed? It will be undergoing the reduction oxidation process. H plus and electrons will be removed. And who will be the cleaver? That is the question. Who will be cleaving this particular molecule? It is nothing but the HSCoA. So here HSCoA is involved. Okay, so let us see what is happening. So HSCoA will be coming. Coenzyme A is coming, and this coenzyme A will be removing this particular carbon dioxide. So this carbon dioxide is removed now. Now this sulfur will be added there. Okay, now we can see that there was a hydrogen H plus here and there is an H here. Okay, now this one will be resulting in the formation of NADH. Okay, so we can just see the NADH. So NAD plus is now forming NAD. So blue H plus H plus. Normally we will not be representing here this particular H plus. In reference books you can just see only the NADH, no NADH plus H plus. Reason is only this hydrogen will be reducing this particular NAD plus, even though this component is actually there. Clear? Now as a result this will be forming succinyl coenzyme A. So the resulting molecule is succinyl CoA. So what will be the, instead of this CO2, S-CoA will be the, so S coenzyme A. Now what is remaining there? C double bond O, CH2, CH2, COO minus. succinyl coenzyme A is now formed. So by release of this CO2, the entire glucose oxidation is over. And whatever remaining reaction that is happening in Krebs cycle is just to retrieve the oxaloacetate that was entered into the Krebs cycle at the first step. So it's just for releasing that oxaloacetate back. Okay, freeing that oxaloacetate back so that we can just start the second cycle of Krebs cycle. And for that purpose only the remaining reaction will be happening. By the formation of succinyl coenzyme A, the oxidation of glucose is over. But you are learning Krebs cycle, so you have to learn till the oxaloacetate is taken back, retrieved. Clear? So hope you understood this particular reaction. So this one will be nothing but the dehydrogenase alpha ketoglutarate dehydrogenase enzyme will be catalyzing this particular reaction. Okay, so now let us see what happens to this succinyl coenzyme A. So next reaction. So make sure that you are drawing this in this way with a different color and all so that you will understand what is happening.